So I'd like to thank Dudley for the introduction and OFAS and AGE for inviting me to give a talk today. And I'm going to give a talk, I should probably introduce that, the benefits of isoleucine restriction on lifespan and health, mainly in mice. So pretty much everyone here, I'm sure, is aware that calorie and protein restriction can extend lifespan and health span, at least in some species, but also that terms and conditions do apply to these interventions. So we know that calorie restriction is universally beneficial. Luigi just gave an excellent introduction, so I don't have to do much work. But basically, it's beneficial in a lot of species. It promotes longevity and health span. In rodents, non-human primates, and humans, it promotes adiposity loss and improves glycemic control. And it reduces many age-associated diseases. Here I'm going to plug mine and Dudley and Luigi's review if you want to read more. But there are some negative impacts. So feeling of hunger, which no one enjoys, feeling cold. It also lowers metabolic rate. So to get a consistent effect, you have to keep lowering calories more and more, which is not ideal. And also bone mass loss and frailty with age. So we were interested in protein restriction, which again, Luigi has already introduced, which is great, which recapitulates many of the benefits of a calorie restricted diet. So this is a study done by the lab a few years ago before I started with Luigi and Nicole Cummings, who's now Nicole Richardson. And basically what they found was protein restriction reduces body weight gain, and it also increases energy expenditure and improves glycemic control. The best part is that it does all this with increased food intake, which is something that I find very appealing in a nutritional intervention. Just a caveat, this is actually a picture of a hamster, not a mouse. So just suspend your disbelief for these slides. I was only recently told. And again, this has already been introduced, it's very repetitive, but in males with a BMI of 30, a short-term clinical trial of a low-protein diet, reduced body weight, reduced fat mass, and improved fasting blood glucose. And there's been many other sort of long-term retrospective and prospective cohort analyses where they look at food intake. And they basically found that if you're in the lower end of the spectrum in terms of eating protein, you tend to have reduced cancer and mortality. And if you're at the higher end, it can double your risk of diabetes and cardiovascular mortality. So it's a pretty powerful intervention. What our lab wanted to do a few years ago, and this was headed up by Dae-Yang Yu and Nicole Richardson, who both finished their PhDs and have gone on to postdocs. They wanted to drill down into what about protein reduction is driving these responses. So they split all the branched chain amino acids up into their separate components. So leucine, isoleucine, and valine, and total branched chain amino acids. And what they found is that isoleucine was a really powerful intervention when it's restricted to improve glycemic control. You can see that in sort of giant here. Even more powerful actually than all the branched chain amino acids together. And you can see here this reduction in body weight. The low isoleucine is the red bar. So something I'm really interested in is genetic background and sex effects on nutritional intervention. So I was really interested in isoleucine, and so I started a set of experiments, and I used Het3 mice. And if you're not familiar with Het3 mice, they are used by the NIA to test their interventions, and they're actually bred from 4 different strains. And so they're all genetically heterogeneous, and they come out like puppies, and they're all different colors, different starting weights. It's very, very cute. But also they're in my opinion, a slightly better model to look at humans because we're also genetically diverse. So it works, then it maybe is a bit more robust. And so I lowered protein in these animals and found that in the males, they had reduced body weight, but in the females, they actually didn't show any changes. And the males, but not the females, had improved glycemic control. So I started these same mice on a low isoleucine regimen. So we had our control mice, which were on 21% calories from protein or amino acids, low amino acid, which is where we reduce it by 2/3, and low isoleucine, where we just reduce the isoleucine, and then we make up for that by increasing non-essential amino acids. So they're getting the same amount of amino acids, but reduced isoleucine only. And so we put them on these diets at 9 weeks old for 12 weeks, and we measured lots of different phenotypes, and this is what we found. So the males had a really profound decrease in body weight on the low isoleucine diet, which was completely not apparent in the low amino acid group, which was really surprising and impressive. And so that was amazing. And they maintained that over the 12 weeks of study. And you can see it's a little difficult, but this far right bar, you can see that the mice are actually much leaner, a bit leaner. That's sort of indicated here. They reduce lean mass and they reduce fat mass, but they become leaner overall. And again, this is despite an increase in food intake in the low isoleucine group. So they are eating more. And we just wanted to make sure that they weren't kind of making up for it with protein. But you can see in the middle graph there that the low amino acid group are eating significantly less protein. And the lower isoleucine group are also eating significantly less isoleucine. In the females, we saw something similar, a bit of a less strong effect in the females, and they gained a bit more weight over time, but still a pretty impressive decrease in body weight, which we hadn't actually seen in our natural source low protein diets before. And you can see they didn't gain any fat mass over time, which is pretty cool and ideal. I added the dots here just so you can see how variable the Het3 mice are. They are heterogeneous, can be annoying, but this is just how it looks. And again, increase in food intake, like the males. We think that this weight loss is partially driven by an increase in energy expenditure. So you can see in both the males and females on the low isoleucine diet, they have an increase in energy expenditure. And that's actually in the low amino acid as well as the low isoleucine group. I think it looks really cool when you look at it over 24 hours of the day. So mice are active at night. So the gray area is the nighttime for the mice, party time. And you can see that they have this really big shift at nighttime, basically, to have much greater energy expenditure over time. And that's even more sustained in the low isoleucine group than the low amino acid group. And you can see that in females as well. It's a bit less impressive, but it's definitely there. We found in the males and the females, they both had an increase in glucose tolerance test. So they had improved glucose tolerance. If you don't know what that is, you increase the amount of glucose and then you measure its blood every 50 minutes. And the quicker they're able to clear it, the better that glucose tolerance is. Interestingly, the low amino acid group in the females did just as well as the isoleucine, but that wasn't the case in the males, where only the low isoleucine group was significantly lower. And this was independent of changes in insulin sensitivity. So that was pretty interesting and a little surprising as well. So I put all the phenotypes I measured together just to kind of get a broad overview of what's going on. So what it looks like is that the male controls and the control and the low isoleucine diets seem to be very far apart, quite far apart, with this low amino acid group in the middle. And if you look at sex, there's quite a big sexually dimorphic response. In general, the females tend to stick a bit close together, even on these different diets. They have a bit less of an impact on the diet, whereas the males are very spread out. And I hope you can see this, but basically what it's showing is a correlated calorie intake, amino acid intake, and isoleucine intake with all the phenotypes. And basically what you can see is that calorie intake and isoleucine intake are the complete inverse of each other. So a high calorie and low isoleucine seems to improve energy expenditure and then a low calorie, high isoleucine diet seems to be very bad for your glucose tolerance. So the next thing I wanted to do was see if this had an impact on frailty and lifespan. Oh, time is moving quickly. So we put these mice on diet at 6 months old to try and get rid of any effect of development on the mice. And so we sacked a cohort after 18 months of diet, so 24 months of age. And we did a lot of these interventions again. And so what we found was that there was a sustained body weight loss on low isoleucine and low amino acid diets when you start them at older ages. And you'll see at 24 months, we get this like funny result here. That's because the control mice started dying and only the thin ones were left. That's why that's a weird artifact there. And we also saw improvements in glycemic control in the males and in the females, but a much stronger effect in the males again, as we would expect. Ooh. And what's cool is, so there's this big study called the Survey of Health of Wisconsin, where they've got about 3,000 or so participants. And they get food diaries and they can see how much they eat. So this is isoleucine percentage relative to protein. So it's not to do with protein intake, it's just to do with isoleucine as a percentage of what you eat. And you can see that if you reduce your isoleucine intake by just 1%, you drop about 3 BMI values, which is pretty impressive. It's a really cool data set and anyone can use it if you want to. So this is the website. I'm not paid by Survey Health of Wisconsin, just FYI. So frailty, we use frailty, which is actually from the Howlett lab. Susan's going to be presenting next, so she can probably tell you more about that. But it's a non-invasive measure of healthiness. There is some human error. We did have a change in researcher because it was COVID. The new researcher was me, and I am a harsh marker, apparently. And, and about half of the mice were sacrificed at about 24 months. But these are all of the frailty things that we measure. So it's everything from loss of whiskers to fur condition to getting like a little back hump. And what we found was that the low amino acid and the low isoleucine diets were able to improve frailty in the older mice, in the males, but not in the females. And this mainly seemed to do with physical frailty. That seemed to be the main driver of this frailty difference. So that's things like body condition, and the little hump, like I was saying, and some other measures. And we also found that urinary dysfunction gets worse as they age, and the low amino acid diets and the low isoleucine diets seem to completely get rid of that. And in the females, it's not really an issue, so that didn't really change. I like to think that long-term memory improved, but as you can see, these error bars are huge, and this is just me hoping and wishing. But I mean, maybe there's something there. I just needed more mice. And we actually did this a bit too early, probably should have done it a bit later when it's more likely they would have memory deficits. A cool thing, I think, even though this has no error bars, no significance bars, is that the low protein and low isoleucine mice didn't seem to have, didn't have muscle frailty as tested by an inverted cling test. And that's something that's really important, because as we age, it's not really helpful if you are alive, if you're just sort of dragging yourself from place to place. So that's good. And we did find an increase in lifespan in the males. So there was a 33% median lifespan increase in the mice on the low isoleucine diet, but no change on the amino acid diet. So that's awesome, and what we were looking for, really. But no change in the females. That's a lie. They had a 7% increase in median lifespan on the low isoleucine diet and nothing on the low protein. So it's a really amazing intervention. And this is a slightly weird graph, but we had one really long-lived control animal that sort of ruined it. So I'd like to put this up. And you can see this is the 10% longest-lived animals for each group. And so you can see that control versus low isoleucine, the mice tended to live about 200 or so more days on the low isoleucine group. And low amino acid and low isoleucine seem to have an increased lifespan as well in the females. Oh, and that's just the p-values. So what's driving this increase in lifespan? We don't really know. So Crystal, who's presenting tomorrow, recently put a paper out that FGF21 is required for these improvements in lifespan on a low protein diet. I'm sorry if I don't think she's talking about this, so I don't think I ruined it. It's published. So, but you can see that the effective lifespan disappears when you knock out FGF21. So naturally, we looked at FGF21. Circulating FGF21 doesn't seem to be any different in the old mice. So that's FGF21 protein in the blood. FGF21 expression in the liver does seem to trend upwards in the males, in the young and the old mice, but there was no effect in females. It's possible I just need to increase my N value for this to make it. But another potential is that FGF21 is produced in brown adipose tissue and muscle as well. So it might be that it's acting in other tissues and not necessarily solely in the liver. We also did some transcriptomics, which and found that in the males on a low isoleucine diet, PPAR signaling was up, peroxisome was up, fatty acid degradation and metabolism was up. And that was really interesting. So liver PPAR alpha is crucial for whole body fatty acid homeostasis. And so it's possible that through this increased turnover of fatty acids, they're reducing the amount of kind of inflammation and oxidative species that are floating around. And in general, that's just causing better health. We did lipidomics, and we also saw there was a greater movement of fatty acids in the lipidomics in the liver as well. The reason I think the fatty acid thing is really interesting, so this is the chamber data from the long-term animals, and you can just see kind of overall, they have greater energy expenditure like before, and they tend to go into, in the REI, you can see they are in lipogenesis for a lot longer and more sustained than the other groups. And it just seems like they're having these more extreme rhythms than the control mice. It's not related to activity levels in the middle there. There was potentially related to this cholinergic synapse. I don't think they're in the liver, but it does seem like there is some sort of relationship between choline possibly produced in the liver and circadian rhythms. And so if you look kind of in the literature, it's related to obesity and circadian rhythms and things like that. This is a complete sidebar and I am running out of time, but COVID did come out in my low isoleucine mice as significantly downregulated in females on a low protein isoleucine diet. That's really just to tell you that KEGG actually does update their pathways every now and then. So that's nice to know. So conclusions from this. So mice with the heterogeneous background, the low isoleucine diet is a super powerful tool to reduce weight gain and improve glycemic control in old and young mice. It reduces frailty and increases lifespan, but only in males. And it's associated with, isoleucine intake is associated with increased BMI in humans. Also, it might promote metabolic flexibility and diurnal rhythms. That's kind of what I'm trying to do next. So we want to integrate the transcriptomics, metabolomics, and lipidomics to determine these changes. One other thing is that because they're so heterogeneous, the pathways were a bit of a nightmare. So we're working with some statisticians to look at individual mouse pathways and try and make some connections that way. Finally, what should you or shouldn't you be eating? Isoleucine is high in everything good: meat, eggs, cheese, nuts. So what you could eat more of is fruit and vegetables. And if you really can't give up your steak, you could consider, low isoleucine is low in coffee though. A coffee is low in isoleucine. But you could also give up your regular steak for an emu steak, which is also very low in isoleucine if you're interested. So just like to thank the Lambing Lab, particularly the wonderful undergrads that helped with this work, Jessie and Kat and Annalisa and Grace, who are wonderful. Everyone in the lab, Young, Michaela and Mariah, all presenting posters, and that's them. And the star of the lab who made it here just in the nick of time, Dudley. Thank you.
Hi, Tim Mackey, University of Washington. You said that the isoleucine restricted mice have increased energy expenditure. Is that due to more activity? Are they more active?
I should have mentioned that. That's the FGF21 link. FGF21 does increase energy expenditure, but we didn't really see massive increases. It's not to do with activity. So we measured the activity and it doesn't really seem to be related. It might be related. Oh, is it here? Here? What? Oh, I can see myself. It's possible that there's increases in maybe browning of the white adipose tissue or something like that that's increasing energy expenditure, and that's sort of my next job.
Okay, cool.